about transcriptomes, replication, and de novo CNVs and how they all tie together in a very interesting story. So take it away.
All right, thank you very much. So yes, I agree with the sunlight, and I didn't touch Ryan because he tells me he's sick, so I'm not going to do that. So this is my long title, Integrating Transcription, Replication, and De Novo CNV Data, which is meant to sound like bioinformatics, because hopefully we'll get to something you all can sink your teeth into, and mechanisms of genome instability. So that's a very long title, so here's my short title, When Worlds Collide, and my high-impact visual for you to appreciate why we were waiting to start. So what do I mean by when worlds collide? So this is what you were taught in school. And for example, if you take Human Genetics 541, which I now direct, you had one day that you came in and we told you all about this process of transcription, which starts at a promoter, and engages in processive synthesis of RNA, copying one of two strands. And then you took an exam, and then you came back in another session and you learned all about replication and the bidirectional spawning of helicases moving in opposite directions, and leading and lagging strands and all these wonderful things we learned about. And then you took the exam and everybody was happy. But what we were afraid to tell you is that these things all have to happen in the same nucleus on the same template of DNA, and that creates problems for the cell and that it has to be able to deal with. And that I think you can appreciate the title then, When Worlds Collide. Now, I have to recognize that there was a pretend marine biologist who was actually the first person to recognize the problem of worlds colliding. And he had the famous line, "A George divided itself cannot stand." And if we simply change that to genome, I think you can appreciate the problems. That this is a very prescient analysis in the 90s. So, okay, so with that as a way of introduction to the general topic we're going to be getting at, I'm going to tell you the following stories today. First, I'm going to tell you a story which is a replication story, and it's a story about copy number variant formation in the human and mouse genome. Then we're going to switch for a time period which is unfortunately going to be too short in the talk. I'm not going to have time to do justice to the BRUCE-seq nascent RNA sequencing project, but a transcription-dependent story. And then we're going to have these worlds collide in the description of a genome research paper which came out online at the end of last year and in press sometime in the last month. And this is going to be the lion's share of what we'll be talking about to lead you through the more bioinformatic analyses in that paper, and then ultimately think about what we've learned in this and its implications for human disease and cancer. So, okay, with that outline, let's have a little bit of introduction on copy number variants. And in this audience, especially with Ryan present, I imagine that you've heard a lot about copy number variants, but nonetheless, let's try to set the stage for specifically how we think about certain parts of this problem. So what is a copy number variant? Well, in the golden era when microarrays were enabling the first passes of genome-wide analyses of, for example, two randomly selected human individuals, Craig Venter and Jim Watson, we could realize that there were regions of the genome in which they didn't have the same copy number. And that, in fact, this proves to be very common in the human genome, so that we see these kilobase to megabase gains and losses at high rates, and there's tens of thousands. And I started putting an X here because I'm tired of trying to keep track of Ryan and how many of these things actually exist in the human genome at this point. And sort of commensurate with that is this fact that there's a reasonably high 1 to 3% mutation rate in meioses. So this is a substantial cause of variance in the human genome. And of course, because they're large, they're affecting a lot of the genome. And I'll emphasize in a minute that these are important for human disease biology. But to set the stage for that, we need to think briefly about the fact that there are two main flavors of copy number variants, so-called recurrent and non-recurrent copy number variants. And I want to just briefly explain what we mean by these terminologies because they certainly can get confusing. Recurrent CNVs are those which occur between two segmental duplications or extended homology repeats within the genome. And so by a process typically non-allelic homologous recombination in meiosis, these two large blocks of homology drive a recombination event. And the reason we call these recurrent is because when you sequence or look at the final product that arose in a series of different individuals, the mutation result is exactly the same. You could not tell them apart, one individual or one mutation to the next, because they're occurring through large blocks of homology. Contrast that with today's focus on non-recurrent CNVs, which are effectively defined by the fact that the endpoints of the copy number variants are dispersed. That as we look at 1 or 2 or 3 individuals, they will each have distinct and distinguishable mutations arising through processes, which we'll talk more about in a little bit. But because these are non-recurrent does not mean that they can't be occurring repeatedly in the same locus of DNA. So in fact, we do, in fact, what today's topic is going to be substantially about is it's difficult, and Martin's going to laugh at me, because it's very hard not to call these recurrent non-recurrent, but they're repeated non-recurrent CNVs that are arising. And here's one example in the AUDS2 gene, which is just a nice review of the myriad of AUDS2 copy number variants that have been observed in human individuals and associated with autism spectrum disorder, intellectual disability, and the like. And again, just at a glance, you can tell that these are all different, distinct, private, if you will, mutations that arose, but are nonetheless all within this gene. And because of the nature of the patient population, of course, there may be a selective aspect to this. So that's in the germline. These are all inherited in the germline. There's also the acquisition of this category of mutations in the somatic lineage. In particular, we're going to think about cancer here briefly. So this has been an ongoing analysis in cancer for quite some time, analyzing. This is work from Sami Malek here at Michigan, looking at CLL, where he characterized what early on were not called copy number variants. They were just deletions of the genome. And all the blue lines here indicate spans of deletion. But again, you can appreciate that as you go down the line, they're all different and non-recurrent deletions that are nonetheless overlapping in for certain regions of the genome, which are then, of course, assumed to have functional importance. So both in the germline and in the somatic lineage, we can see clear evidence of this process of repeated non-recurrent CNV formation that has clear implications for human health. So we want to understand something about the mechanisms behind this process. So I do this, I'm going to tell you these couple of stories. And the first story is the one that starts with work from Martin Arlt, who's now a research track faculty member working in Tom Glover's laboratory. And Martin was the one who really established the cell system I'm going to tell you about on the next slide. Irene Park and others in the lab have contributed in ways we'll try to mention as we go along. So what Martin and Tom developed and had done before I began this now very long collaboration working with them on this project, is to establish a cell system in which normal cells, in this case normal human fibroblasts, so karyotypically normal, turd immortalized in most of the studies we're going to be talking about, are grown in culture and treated with various agents and allowed to recover and then plated for single clones, which are then expanded and subjected to relatively unbiased genomic analysis. When I say relatively unbiased, what I mean is we don't believe there's a lot of phenotypic selection going on. Events are typically going to be heterozygous. Obviously, some things might be deleterious and lost. The agents we're going to treat them with are largely going to be replication inhibitors, which is where we get to the replication link in this story. Acetylcholine is a DNA polymerase inhibitor. Hydroxyurea is a ribonucleotide reductase inhibitor, and so is an indirect inhibitor of replication by altering nucleotide pools in the cell. So we go through this process, we establish clones, and we analyze them for their CNV content. And in the studies I'm going to be telling you today, almost all of the data has been generated by SNP or array CGH microarrays. So this is a slide to just show you what these look like. So I've overplotted in magenta a cell clone that was analyzed compared to in blue its parental cell clone. So just it makes it very easy to see that there's a de novo CNV that formed in the clone that was not present in its parent. One of the main reasons I show this slide is that it's very easy to find that. This is a bioinformatics group. Finding these in the bioinformatics data would not stress even any one of you. This is very easy, and there's a reason for that, and that is that these are fairly large, okay? I also will, just before I make that point, I'll just emphasize that this occurred in a gene, and if you're thinking about the scale here, that's a pretty big gene, and so keep that in mind as we move along. So I said these are easy to find because they're pretty big, and this is a slide to help just briefly talk through the issue that indeed we don't believe that this is a detection bias, that this really is the size distribution of these is fairly large, with the median size about in this study, in this slide at the time it was presented, 186 kb, but generally in most studies hovering around that 200 kilobase mark. And the reason I want to just emphasize that we don't think it's a selection bias. Of course, we do have a lower detection limit in the microarrays. But first of all, we're seeing a clear drop-off in frequency above the threshold of where we actually can detect CMV. So that's point number one. And also, we have analyzed a subset of these clones by next-generation sequencing, and we didn't see the appearance of a whole large class of much smaller events in these clones that we analyzed. And so we don't really think we're missing that many in this lower size range. And for those of you, I don't have it plotted here, but if I were to plot population CNV, they don't show this pattern. They show a different pattern where, in fact, things become more and more frequent as you get to smaller events. So this isn't the same as what you see in the germline. But again, these are relatively unselected in the cell experiment system. They are, however, very typical of the size range in which you see with pathogenic CNV, which, of course, is why we're interested. All right, so I'm going to tell you other things about these CNVs. The first one is sort of been implicit in what I've been saying, but I'll just say it out loud. They are induced by these replication stressors. So we see a non-zero rate of CNV formation with untreated cells. So simply growing cells in culture, you will see the acquisition of de novo copy number variants. But the frequency is clearly increased with both the phytocolin and hydroxyurea-based replication stress. And that's a lot of work, mostly from Martin on this slide, and it's done now. So, sorry, Martin. So the other thing I need to tell you about these is that, like essentially all of the CNVs in this non-recurrent class, what we see at the junctions is little or no micro-homology, but no extended homology. So this, for example, is a blunt-ended junction. Here are two short micro-homology junctions. This is a small insertion. This is, for people, when we do this, this is garden variety. It's sort of a running joke in the lab. Let me guess, what did you see? Microhomology. This is so boring after a while. This is what you see in human germline variants, which is what you see in pathogenic variants, and it's what you see in ours. And it establishes this idea that, first of all, this is part of why they're the non-recurrent class, but also suggests that they're arising by non-homologous repair mechanisms. So what does that mean? We're not going to spend a lot of time thinking about repair mechanisms. That's not really the subject for today's talk. But let me just first of all tell you that you can imagine this happening by an end-to-end rejoining process where breaks are being rejoined by mismatching ends and just, as we say in the end joining field, sticking the ends back together. We don't actually believe this is the predominant thing going on, and I'm going to show you two reasons on this slide. The first here is just to summarize, again, one of Martin's elegant long studies that we tested this, and they don't require XOCC4, which is a non-homologous end joining gene. And so it doesn't appear as though they're going through that pathway. The other thing that we can see, though, is the occasional appearance of what we're going to call complex CNVs. This is just one example that was analyzed by both microarray as well as sequencing here. And without going through the details of the slide, we can see that this event can be described as a continuity of the genome that proceeds in this direction, has a junction that jumps here, copies that in the reverse direction, has a second inversion junction, and then continues on. So there are two junctions in a single event in this particular CNV. And as a result of that, it's hard to describe that by an end joining process, but it's relatively easy to describe it by a template switching process in which a strand has moved, for example, from a replication fork and literally jumped to a different replication fork. And if we simply allow that process to repeat, we could easily form two junctions within a single CNV. These are ideas principally described by Jim Lepsky and colleagues. So that's all I'm really going to say about the sort of DNA strand-based mechanism. The rest of the talk is going to be driven at the question of why would this be happening? What are the substrates, and how can you describe where these things might be coming from? And to get there, we're going to have to think about yet one more property of these CNVs, and that is where they are in the genome and what their distribution in the genome is. So our CNV sets, by our standards, are huge because we work very hard to identify any one of these in our experimental cell system. By human genomic standards, these are, of course, small numbers compared to the many thousands that you could study. But this is what we've got. And when we spray them around, we look at where they are in the genome. We can see, first of all, that they occur everywhere. There doesn't seem to be a particular chromosome or anything that's missing. And there are singletons and so on. But then there are obviously other loci in the genome where they're clearly clustering. And I'll show you this in more detail on the next slide. Now, to a mutagenesis scientist like myself, this concept of having, of querying mutations and then seeing that they occur in a lot of places, but that there are certain hotspots. This is what every mutagenesis experiment in the world looks like. But the question is, what is the basis of that hotspot? Now, one of the things that's true is that most hotspot type mutagenesis assays are looking at a very small and restricted scale, often to the base pair, this base. And in fact, you can see these signatures of non-random mutagenesis happening in the human genome at a base pair level. But these, I want to emphasize, are not, they're a much, much larger scale phenomenon. Our CNVs themselves are hundreds of kilobases, right? So the clustering of these occurs over the scale of 1 to 2 megabases of the genome. So this is what we always, the words we like to use to describe this is a regional hotspot of the genome. This is a very large chunk of DNA, bigger than the chromosomes of many species. Okay, so that's what we're seeing. Now, what might be describing this regional property? Well, my argument has always been we need to look for something that is itself big. Well, the obvious candidate is genes. Genes can be big. So is there a correspondence of these two genes? Here we're showing one of our hotspots in the AUTS2 gene, which I mentioned earlier. So indeed, much like the human patients, we see a clustering of CNVs, which show a very nice correspondence to the AUTS2 gene. So that's very interesting and very exciting. But we also see other hotspots, like this one at 3q13, which is actually the hottest spot in our fibroblast cell data. And it's adjacent to this 600 kilobase gene called LSAMP, but the bulk of the CNVs do not appear to occur in that gene. And so that was one of the types of data sets that gave us pause as to how good this correlation with genes really was. Now, the other thing I need to tell you about is a different manifestation of this clustering of chromosomal instability events. And this is the phenomenon that actually led Tom Glover to do these studies in the first place, was the phenomenon of common fragile sites. So what is a fragile site? So you take cells, you treat them with replication stresses like acetylcholine. And what you do is you make metaphase spreads of chromosomes, and you can observe, if you're Tom or Martin, if not if you're me, you can actually say, that's chromosome 3, and in fact, I know which band of chromosome 3 that is, and you can tell that it happens repeatedly in the same place in the chromosome. And FRA16b and 3b are the two of the hottest spots in the lymphoblast cell types that these studies are typically done in. So the idea is that the copy number variants might be associated with these self-same sites of preferred breakage. So is that true? Well, FRA16b in gene WWOX, a large 1.1 megabase gene, in fact, it is true. We do have a CNV hotspot at that locus. However, FRA3b in the large FIT gene is not a site of CNV formation in our studies. And if you maybe paying attention here, you'll realize that that's chromosome 3 and that's chromosome 3. Well, this cluster on chromosome 3 had not been described at the time that these studies were initially being presented as a chromosome fragile site. So we had clear exceptions to the rule that were really quite bothersome in trying to figure out what these connections were. So the secret to that was Matzah Jungman. So here's the second part of the talk, is thinking with through the studies that I've been doing with Matzah, which initially weren't really executed with any thought process of this other thing I was doing with Tom. And this is a part of the sense of worlds colliding, is that we realized one day that we were actually studying the same problem. And so what Matzah's been doing, and that I've been helping on the bioinformatics end of this, is looking at BRUSeq technologies for nascent RNA sequencing. So what is this? And again, I'm going to have far too little time to go through the power of these very elegant techniques. But the concept is this, is that we're going to take bromouridine, which is a ribonucleotide analog, which is going to get incorporated into nascently synthesizing RNA. And we can do various things to this. I'm not going to show you any data. We can actually pulse chase this. But actually, I'm going to show it right here, sorry. So if we pulse chase this, we can actually look at RNAs, which are now 6 hours old. This is somewhat like looking at mature RNAs, except they're not all mature RNAs. It's 6-hour-old RNAs. But they have been processed, which is the point. And so if we look at that signal in magenta, we see that the signal nicely points all up to the exons of this gene, as you would expect. If we, however, harvest that RNA immediately after labeling, what we see is a very different pattern, and the signal is now diffusely distributed throughout the body of the gene without respect for exons and introns because it is nascent RNA. And I choose this example gene on purpose because you'll notice that there is no signal at the supposed first and the annotated first exon of this gene. We do see an exonic signal here, and we see signal in the introns, or the nascent RNA, across the entire span. So we learned something about the transcription of this gene in the cell type that was under study in this particular instance. Now, it would be nice if we could absolutely definitively establish that there were a promoter there. So we have another little trick that, again, all this is sort of Matzah's baby that figured all this out, but that if you take these cells and UV irradiate them immediately prior to the bromouridine exposure, what you do is you create blocks for the progression of RNA polymerase. And if you think about this for long enough, and if you watch Matzah's train analogies, you'll figure out that what's going to happen is that new initiation keeps pushing into these blocks. You will get an accumulation of the signals at the point where transcription is starting. And so we get, this is the same data from the nascent RNA. If we take the nascent RNA with the UV block, we see that it accumulates right at that predicted point of transcription initiation. So that's the basic suite of techniques. And just to give you just a few slides about the flavor of this, a couple of which will set the stage for what we're going to see in the CMB studies. We can do lots of things with this. So one of the things I want to emphasize is that this is a vital process, which is to say we're treating live cells. We don't have to harvest them. We simply take the cells, treat them with the agent, and harvest the RNA from a cell which is living and doing all of its proper stuff. So we can do lots of things like look in induction. In this case, this is an ionizing radiation treatment. And what we can see is the clear and obvious induction. And one of the things that's very nice about this is, first of all, we're looking at transcription, not RNA. It's a subtle difference, but a difference. And we see signal across the entire, integrated across the entire body of a gene. And so we can come to very high degrees of confidence about the changes in the transcription state of a given gene based on that. And this is a threefold change. And I hope you can appreciate with replicates looking like this, we can see much less than a threefold change. We can easily go below twofold in terms of looking at induction state. And I don't think I'm going to show a slide. I think I pulled it out. But you can imagine that based on a timing basis, we could also see how far that transcriptional wave of induction had moved down a gene within a certain period of time. Just as a sort of a reference control, you can see this gene. I like this combination here because that gene immediately next door is not induced at all. So we can expand that to lots of studies. And this is sort of echoing what we're going to see as being important in the CMV studies in just a moment, that we can look at the existence of these isoforms. So here's the actual annotated, RefSeq annotated version of this gene, which I apologize, I forgot to label here. And we can see that there is evidence for a couple of different isoforms which are being differentially utilized. So the long isoform is present in this strain, but is actually much more prominently expressed in the other strain. And this is the kinds of things that we can do on a genomic basis now. That's, I think, much easier than doing with the actual nascent RNA because we look at the transcription itself. And the last slide I'm going to show here is just a little bit of an advertisement for a study that we have submitted, looking at one of the values of this UVA radiation capture technique. So what we basically think is happening is that we're capturing these transcribing RNA polymerases on the DNA by putting this block in front of them. And the consequence of that is that the RNAs that they're transcribing become stabilized if they were going to be highly labile. And one of the really interesting classes for this is enhancer RNAs. So RNAs being transcribed from enhancers, it turns out that we see a potent stabilization or detection of these RNAs. And this is the well-described Phos enhancer, and you can appreciate bidirectional transcription coming out of this enhancer. This is, of course, a particularly nice-looking example, but we nonetheless can analyze this data on a genome-wide basis and have high hopes that you can actually stratify enhancers by their functional utilization by this type of approach. So that's just a few little tidbits of what we're seeing in the BRUSeq data. But now we have to get back to today's main topic and get back to this question of worlds colliding. All right. So what do I mean? Again, to come back to this. So we have this set of sort of connections between genes that we're seeing, copy number variants, and common fragile sites, but it's been a little bit murky. And so the answer to this is going to be transcription, perhaps not surprisingly because genes are transcribed, but nonetheless, this is something that has to be demonstrated. All right, so here's the demonstration, and the titles of this slide is in red because if there's one thing you remember from today's talk, it's in red, that the CMV hotspots and then common fragile sites correspond to large transcribed genes. This is our central observation. And again, in some ways it's obvious, but it needs to be demonstrated that, in fact, it is the transcription of these genes that's the critical property. So I'm going to give you all sorts of evidence, but let's start with anecdotally just a couple of first examples, some of the hotter spots in mouse and human. And also, I haven't really told you about our studies in mouse embryonic stem cells, but we've done this in more than one species and more than one cell type. So if we look at the clustering of CMVs, and then we look at the transcriptional profile, where I didn't mention this yet, but negative deflections on the curve mean that they are transcribing in the reverse direction, and negative deflection on the CMV scale reflects that they are deletions, not duplications or amplifications. And what we see is that there is a nice correspondence of this population of CMVs with that transcription region. Same thing here. And this is actually replication data. And for now, you can either look at it or ignore it, your preference. But we'll talk more about the replication part of this in a moment. What I want you to think about briefly, though, is just the annotations down here. And what I can tell you is that when we started these studies, a lot of these, or not a lot, some of these long spans of transcription. And when I say long, you got to understand, we're talking about megabases here. These are 2 megabase-ish sizes of transcription. And some of them were not annotated. So at the time that we prepared this slide, the ensemble annotation still did not have the annotation of this very much longer isoform of this otherwise very small gene, beta-3-GALT1. It actually is in the annotation now. And the same thing happened here, that LSAMP, a gene I mentioned earlier, and I told you it was 600 kilobases. Well, we discovered, as did others apparently, since it entered the ensemble annotation, that in fact there is an isoform of this gene which adds, I forget exactly how big the intron is, but it's something like a 1.6 megabase intron. Really huge expansions of the range of transcription. And with BRUCIC, we can demonstrate to you it really is transcribing through that entire region contiguously. Okay? So that's the general phenomenon that we're talking about. So let's make this a little bit more rigorous for a bioinformatics audience. So what I'm going to do is to lead you through some of the sort of more systematic and global analyses of this data set. And to do so, we have to just agree on a couple of simple definitions. When I say transcription unit, I'm referring to this contiguous span of transcription that we can detect by BRUCIC. So it's not a hypothetical. We know that we see transcription going from here to here. And we call that a transcription unit. They're strand specific. So in fact, this particular one, we can actually document the existence of a transcription unit on either strand. And when I say CNV regions, we're talking about this cluster of overlapping or closely adjacent CNVs, and this bothered our reviewer. This was somewhat arbitrary, but I can tell you it doesn't really matter what we choose. And this might seem like a very large number, that CNVs that were closer than 750 kb, that's a long way. But remember, the regions we're querying are megabases in size. So this is not really a problem for the data. So that's what CMV regions and transcription units are. So here's a simulation analysis, which is plotted in a somewhat busy sort of a way. So I'm going to lead you carefully through this slide to think with me about what you can see in this data set. So first of all, we're going to take our CMV regions and we're going to perform a stratification by the number of CMVs that they contain. And the reason we're going to do this is because we're interested in determining whether singleton CNVs versus clusters of CNVs might reflect different kinds of properties of the genome. So that's the first thing we're going to do. Then we're going to simply point out that you can count the number of such regions that we have at the top there. So there are, for example, 9 hotspots. And I didn't mention this. This cluster of greater than equal 5 was determined by a simulation process which allowed us to assert that 5 or more CNVs in our data set with the number of CNVs that we had was a non-random cluster. So we have 9 such of those clusters in our data set. On the y-axis, we're going to plot all manner of different assessments that we're going to do in trying to compare CNVs and transcription units. There are, if you don't think I show enough in the talk, go to the paper. You'll see more of these plots than you know what to do with. I'm going to show you just a couple of the key ones here. And one of my favorite ways of thinking about this was to look at the overlap between the CNV region and the transcription unit as a function of the percentage or fraction of the CNV region that corresponded to a transcription unit. And just briefly, this is actually a more strict criteria than simple binary overlap, because that could be 2 two elements crossing at their edges. What we're really asking is that our CNV regions really occupied by transcription units? And we're going to see, you'll see that the answer is yes. The red is the actual region average of all of the events. And so it's a way of just aggregating all of them. We can also plot them all individually. And then the last part in the box and whiskers is the 10,000 iteration random simulation, where all of these CNV regions were distributed randomly and iteratively about the genome, just to see what we would expect by a so-called random chance. And what we observe now with all of this is that we clearly see, first of all, an enrichment of transcription units in these CNV regions. They are transcribed at a disproportionately high level. And that's true for all of them, singletons as well as hotspots. Okay? However, you can judge by the slope of this that the phenomenon becomes more and more important as you move toward larger and larger transcription units. They are increasingly occupied by these transcription and transcription units. Okay? So I've given you the hint along the way that large genes is what's really going on here, that these are big genes. And in fact, that's part of why we can see this very high degree of occupancy, because there's such a strong correspondence between these large CNVs and large genes. So if I simply take that same plot and now stratify it by, not stratify, but score it as crossing with transcription units in excess of half a megabase, 500 kb, what you see is that the same phenomenon is existent, but the degree of enrichment for the singletons is drifting toward actually the random distribution. It's becoming less important. Whereas with the hotspots, it's becoming actually, and still more important, than the slope is increasing, which basically is telling you that the large genes are really what's driving this particular set of hotspots. Transcription is associated with all of them, but large gene transcription is associated with the hotspots preferentially. So to look at that in a little more detail, we're going to do sort of the reciprocal analysis, and rather than randomizing the CNV region positions, we're going to randomize the transcription units and keep the CNV regions fixed. It's somewhat the same, you expect to get largely the same answer, but it allows us to now look and make a plot of the degree of enrichment as a function of transcription unit length. Same kind of plot, and what you can see is that it really is a strongly length-dependent phenomenon in terms of the correspondence of transcribed regions of the genome to CNVs. And I might argue that this is something of an exponential increase, which we'll come back to briefly later. And the other way of just thinking about this is that at that data point, the 12 transcription units, and again, this is actual transcribed regions of the DNA, greater than 1 megabase, 11 out of the 12 of them yielded CNVs in our relatively small data set. So we think that's fairly striking. Okay, so based on that, we said we believe we can predict the locations of where CNVs will occur based on where large genes are being transcribed. So is that true? So to do this, we're going to now make use of another normal human fibroblast cell line. So the data I've been telling you about have been executed principally on a cell line called 090. It's a cell line that we use, not that many other people use it. HF1 is the fibroblast cell line that Matzah has used in most of his BRUCE-seq validation studies. So we now have the opportunity to compare two cell lines. So we obtained BRUCE-seq analysis on 090, and now we said, having not yet done the CNV analysis, can we predict where we might see novel CNV formation in HF1? So let me walk you through this then. There's a lot of busy data on this table, but it's relatively easy to digest. That when we look at the transcript, the transcription units in excess of 1 megabase that are present in both of the cell lines, we see CNVs in both of them. When we look at the transcripts that are only present in the 090 cell line in the long isoform, we see very significantly an accumulation of CNVs in 090 only, but not in HF1. And this is, I don't have the data up here, the numbers up here, but that's clearly significant by test. Now, the other one, which is what we were really hoping to get after, was actually to observe transcripts that were only present in HF1, but weren't expressed in 090. And there were two of these. Now, I want to emphasize, and I'll do this on the next slide as well, that they are expressed in 090, just not in the long isoform. All right. Now, we didn't detect any CNVs, but this was with a relatively small number of HF1 arrays compared to what we've done with 090. So to be able to ramp this up without breaking our bank, we actually turned to a directed set of assays using SNP assays, so just a simple gel-based RFLP assay. And the left is simply validating a point that I've already made, that at regions that HF1 does not transcribe, it does not show CMVs. But the real kicker on the slide is over here, where we have this gene, DAB1, which you can see has short and long isoforms. 090 expresses the short isoform. HF1 clearly expresses the long isoform. And so we put various SNP probes within here, and we detected 2 CMVs that correspond, as do they all, to the middle of the long isoform transcription, and we only detected them in HF1 and not 090. The p-value is maybe less inspiring than we might have liked, but the way we like to state this result is that we went into a place in the genome that we have never seen a CNV before, and based on the transcriptional profile said we would see CNVs there, and we did. And so we actually think that p-values set them aside. This is actually, it was a fairly robust demonstration of the result. Okay, so what about fragile sites? Sure. All of these are measured in these cells growing in tissue culture. And they are obtained from the exact cells growing in tissue culture. So I have a flask of one cell type, a flask of another, and I harvest their RNA and measure it. Okay. They're fibroblasts. So it's a primary tertimmortalized fibroblast. Actually, other than the ES cells, which I briefly alluded to, that's what all of the cells in these studies are. Okay. All right. So fragile sites was one of our other connections we were trying to make here. What we've observed, if I were to just repeat briefly what we saw on the last slide, is that because we see a transcriptional dependence of this type, we actually imagine that there could be a very strong cell type specificity to these phenomena. And in fact, that now becomes a simple way of explaining all of our conundrums previously. And in fact, it explains the common fragile site conundrum as well. So what we realized is that we had to measure fragile sites in the fibroblasts that we were studying in order to make proper comparisons. And when we do so, I won't lead you through this complicated table, but the data are there, that we see a very good degree of correspondence. You can get a visual impression of this by simply comparing the CNVs and the fragile sites on the top and the bottom. And you can just quickly realize that there's a very strong degree of correspondence between them. And then when I settle on this 3Q13 hotspot, this was one of those times when the literature helped us. We already knew when we read the paper from the Desbatisse group in France that they demonstrated that indeed, not in lymphoblasts, but in fibroblasts, certain fibroblast cell lines will express a fragile site at the 3Q13 locus. So indeed, these do correspond. Our prior exception really was not an exception. Here's another way of looking at the fragile site part of the story, and that is to ask, so we had the very interesting region on chromosome 7 now, where we had 2 genes very close to each other, which showed, in one case, a common long isoform transcription, and in the case of this gene we keep coming back to, AUDS2, a difference in the pattern. So 090 in this case expressed the long isoform, and HF1 expressed, it's a little unimpressive in the way the slide ends up having to appear, because we end up having to expand the scale so far. This is very clear and unquestionable transcription of the well-described short isoform of this gene in HF1. And down here we do just showing just one quick panel to demonstrate FISH analysis, which allows us to ask pointedly whether fragile site breaks were occurring at this gene or that gene. And the bottom line is that in HF1, which did not express the long isoform, we did not see chromosome fragile site breaks. So this, once again, is just another example of just trying to make sure we're right about this, that we do see a nice predictive ability based on the length of the transcription unit. And so this is my slide that you will never forget, that length, when it comes to transcription units, length matters, okay? It's all about how long they are, and I'll briefly mention now and on the next slide that it kind of bothers people that, in fact, intensity of transcription is really not a predictor of where these things occur. It's actually the length of the transcription that matters. So why is that? What's going on here? And now we need to get to back to the replication part. Remember, this is worlds colliding. So what does this have to do with replication? So let me tell you this part of the story, which centers on this technique called RepliSeq, which is very much analogous to BRUCeq, in that this is BRDU labeling to execute nascent DNA sequencing. No one really calls it that, but that's what it is. So what we're going to then do, and as described by Janssen et al., so this is to take the cells that we've labeled in this way and then fractionate them. so that we can now make plots through G1 stages of S and G2 of where nascent DNA synthesis, i.e. replication, was occurring as a function of relative time in the cell cycle. And this is not my slide, and in fact, the data that we're going to be using here is not our data. We actually are using and consuming the ENCODE data here. We did not actually repeat RepliSeq studies in our fibroblasts yet. And there actually turns out to be that this really wasn't necessary, and the reviewers agreed with us. One reason is that replication timing data is much more stable comparing cell lines of a type than is transcription. So that's point number one. And point number two is some of the critical observations I'm going to make on the next slides. I don't need the CMV component at all. And I can actually compare nascent RNA and DNA sequencing from the same cell types without any difficulty by consuming the ENCODE data. So let's show you what I mean. So some of this is things that we certainly know prior to our study, but we add a couple of things to this. One is that we're actually using our BRUSeq actual spans of transcription, what I would argue is a better way of actually measuring transcription than other inferences that can get made. And the other thing is that we can obviously add our CMV data to this. So what we see in the top left is sort of by definition a balanced distribution of replication timing when we consider the whole genome through these different stages, because that's how the study is executed. But if we stratify it by the transcribed in green and the untranscribed in that red color portions of the genome, you can see a very obvious bias that the transcribed genome replicates early, the untranscribed genome replicates late. Yeah, we didn't describe this phenomenon. But what we can now demonstrate is that when we take the same genome and the same data and stratify it by the number of CNVs we see in this region, that these low intensity ones, for example, the singleton CNVs in the red color, skew just a little bit toward early replication, which makes sense because they're transcribed. However, these hotspot CNVs, even though they're transcribed, do something very unusual. They stay or they exist at very late replication profile. Okay? They're transcribed, but they replicate late. And that's unusual on a genome-wide basis. All right, so why is this? It's because they're long. Now, if you're prone to seizures, the next part of the slide might be a time to turn away. So this is an analysis where what we're plotting is replication timing on the x-axis and transcription intensity on the y-axis. And as the animation proceeds, you're moving to larger and larger transcription units. So you're seeing what happens to transcription and replication timing as genes get longer. And what's very obvious is that as genes get longer, they transcribe later and later. This is two different cell types using perfectly paired replication and transcription data, so we have no difficulty with cell type comparison problems here. What I also want you to notice, in addition to the obvious later replication timing, is that the transcriptional intensity, while it's a fairly subtle effect on the slide, this is actually a log-based axis. This state designation is log-based, and it's slowly drifting downwards. And so it's something in the range of about a 5-fold drop in average transcriptional intensity. So long genes are not very highly transcribed. This may not surprise you, but it actually accounts for this fact when I tell you that our CNV intensities don't correlate with transcriptional intensity. It's because long genes aren't really that highly transcribed on the grand scheme of the nucleus. So how do we explain all of that? So here's actually a couple of my favorite plots, but there's some of them more difficult to understand. So let's just quickly try to see if we can't make sense of this. So first, pay attention to the blue and the red traces here, where we're looking at replication timing on the y-axis as a function of transcription unit size. And again, we are talking about measured transcription. And what you can see is that I'm sorry, I need to explain this. Then we're going to plot data from 3 points in genes, the ends of the genes and the middle. And so for now, just look at the ends of the gene. And what I want you to see is that the ends of the genes transcribe later and later as gene size gets bigger. And this is important because we're trying to understand why it is that large genes replicate late. One of the reasons is that their actual replication timing starts later at the end. Is there still a beneficial effect of being transcribed toward earlier replication? Indeed, there is, and this is a little bit subtle, but if you just look here, this is comparing large genes between two cell lines of which only one of them expresses the gene. And what I want you to just quickly notice is that there's no overlap here. And the reason for that is that IMR90, this particular fibroblast line that expresses the genes labeled in red, has pulled them toward slightly earlier replication, and the same thing is happening in reciprocal on the other cell line. And what that basically is telling you is that even the large genes do have this pulling toward earlier replication timing as a result of being transcribed, but it's just much, much less obvious than it is with small genes. Small genes get pulled all the way up to the earliest replication time, okay? So that's what's happening at the ends of the genes. But then another thing I need you to notice is that the middle of the genes, as gene size increases, the middles of the genes become disproportionately later relative to their ends. And this is sort of not obvious maybe the first time you think about it, but it's all depicted right here. If we simply make the assumption that all of the origins that are replicating these regions flank the genes, then the spot in the middle of the gene necessarily becomes farther and farther away from its origin as gene length increases. And that's a perfectly simple way of explaining that observation. It also can be seen in this slide, so I do something fairly standard analysis. We take all these transcription units, line them up, and look at their disposition of the relative replication timing, and we see this obvious U shape to the relative replication timing, which again is that result of the fact that replication is proceeding inward from the ends. All right? Now, the punchline of this is we're always going to keep trying to connect this back to CNVs, and this is what we see when we do that same type of alignment plot with the CNVs. When we look at the position of deletion copy number variants, they correspond precisely to the center point of that latest replication timing, what would actually be then the termination zone of replication at the centers of these large genes. Duplication CNVs don't do that. In fact, they have this tendency to show a reciprocal sort of alignment where they actually are accumulating on the flanks of these large transcription units. Okay. A picture is worth 1,000 words. So that's a lot of graphs and a lot of stuff. Let's just tie all this together and help you understand what we're talking about. All right, so replication is proceeding inward from two ends towards some central region of the genome. And because what we do to these cells is stress them with replication stressors, we see blocks in replication progression. That's the hypothesis. At untranscribed regions of the genome, it's well understood that we can get the firing of late or sometimes called dormant origins, which will rescue the replication of this region. Our hypothesis is that large transcription units change this reality from the genome, and that what's going to happen now is that when these same replication failures occur, that transcription of this very large gene has necessarily proceeded into S phase. And the core hypothesis is that they then evict the pre-replication complexes which may have been bound there, preventing any possibility for firing of these late origins within these large transcription units, and enforcing that reality that replication has to proceed inward through these megabase scale replicons of the genome. As a result, if this happens and there's fork failures on both sides, we have to jump one way or another. And if we jump over this unreplicated gap, we get a deletion. And so they accumulate in the middle of the genes. Whereas if we jump backwards and re-replicate the region, we actually jump away from the ends of the gene. And that's exactly what I showed you we see in the data. All right, we're getting at the end here, so I'm going to skip that little bit of math. Just two more quick slides to demonstrate, to come back to this idea that we do believe this is relevant for human disease biology. I've mentioned AUDS2. This is a much longer list of these long genes which are clearly associated with human syndromic and oftentimes neurological disorders. And lastly, in terms of the cancer genome, what do we see in the cancer genome? So we've begun a collaboration with Ramin Baroukhim at the Broad Institute, who's been feeding us his CMV analyses of the TCGA data. And here's a couple of plots showing you cancer data and our cell line data to show you the clear correspondence of hotspots of CMV formation or copy number alteration formation in cancer in our cells. And we can see that at different sites. The coloring here is reflecting two different cancers, uterine and colon. And just at a glance, you can now tell that this is a cell type or a cancer type specific phenomenon. that not all cancers show the same large gene clustering. And our hypothesis then is that this is predicted by transcription. Because of the story I've been telling you, it also means that we predict that these cells are under replication stress, which is actually a described aspect of cancer cells. And then the last thing I'm just going to quickly show you here is that CDKN2A over here is also a deletion hotspot in cells. But this is the P19 tumor suppressor gene. So you would actually assume that this is the reason for this deletion hotspot is actually functional selection. And that gets to this last issue, which is really the underlying importance of this for genomics, which is that we have to come to a better grips of understanding when hypermutagenicity is a function of selection for mutations and their functional properties versus when it is simply a passenger effect reflecting the fact that certain regions are hypermutable. All right, so the conclusions are stated here. Replication stress induces CMVs. They don't require end joining. They correspond to large transcribed genes and fragile sites, and that we can make a predictive model on this that clearly works in vivo and that we're studying in these other scenarios. Here are the people who did the work, and I've mentioned most of them, and I'm at the end of my time, so I'll stop there.
Open this up for questions.
Yep. So if you go back, and I could go back there, my slide of replication template switching, my first one, clearly invokes the concept of looping. Exactly that. I think that when we talk about this megabase size CMV, the jump almost certainly was much closer in physical proximity. And clearly there's data, there's continuing data to elaborate the idea. Yeah, that replication domains represent I forget what's the letters that people are using to refer to these yeah, the TADs. Thank you. Yes, topologically that they actually are correspondent to each other. So indeed, there's every reason to think that these even though we think of them as coming in from long distances, that they're part of something that's set here in the nucleus. And can we get more precise than that? It's not clear to me, but I certainly think that's part of what's going on here. Mm-hmm. Yeah, I would be interested to go through the data. And I haven't done this, but it'd be interesting to go through and see whether that holds true for the longest genes. Mm-hmm.
So I got a good question, and maybe you covered this and I missed it. Did you so looking at the different cell lines and seeing the different long versus short isoforms, it's pretty convincing evidence. Did you actually try to induce perhaps the longer isoform in a particular cell line and then see after treatment if that
So yes, very acutely asked the question that what we have not done is within everything is correlative between cell lines.
Yeah.
What we want to do and others want to do and we're told are doing is to actually go the other way, which is to silence the long isoform by it's probably easier to take out
as
the promoter.
Induce, yeah.
Because we actually know pretty well where these things start. It's not like this is not an ambiguous phenomenon. It really has a defined start site from a defined promoter, and we just need to use modern genome engineering technologies to go through and knock it out. And we've talked a lot about how we could go about doing this, and we have actually the ability through our deletions to actually know the complete SNP haplotype of these regions of the genome. So I don't actually even need to do this on both alleles. We can just take out the promoter, and it's actually really powerful because then what we predict is we'd only see C information
on that one allele,
on one remaining transcribed allele. So yeah, we need to get that done.
Okay, well, thank you for an excellent talk, Tom.